So content-wise, we have reached the end of the lesson. However, as per usual, we have exam type questions that we need to answer. So let's look at the exam type questions. Let's answer them. Please do pause. Look at the diagram. Look at the question. Write down the answer that you think is correct. And then unpause the video and check if your answer is the same as mine. If you have any differences or any questions, please post the question in the comments and then I will do my best to answer it as soon as possible. But answer it, I will. All right, the first question. Indicate whether each of the statements in column 1 applies to A only, B only, both A and B, or none of the items in column 2. Write A only, B only, both A and B, or none next to the question number 3.1 to 3.3 in the answer book. All right, so this is a match column question. This side, column 1, we will be having descriptions and then we will be given options in column 2. If the description in column 1 matches A, you don't just write A. Please check the instructions. They say write A only, and they've even bolded it. So it's an instruction that we need to follow. So you don't just write A, you write A only. If it does not apply to A, but it applies to B, you write B only. If it applies to both of them, you write both A and B. You don't just write both. You write both A and B. And if it applies to none of them, you merely write none. Okay, guys, very important. Follow the instruction. There's a reason why they have bolded these. Please write as expected. Let's look at the questions. Now, how you answer these type of questions relates to how you answer multiple choice questions, okay? So you read the description and then you eliminate the option that does not apply to the description. You eliminate the option that you are sure does not apply to the description in column 1 to maximize your chances. So the more you eliminate, the better chances of you getting it correct. So even if you don't know the correct answer per se, but when you eliminate the ones that you are definitely sure are incorrect, you are essentially increasing the chances of you getting correct marks. So each answer will be 2 marks. So this carries a lot of marks, guys. So please take your time. 2 marks, 2 minutes. Okay, you have 2 minutes to answer each sub-question. So please take your time. I'm not saying relax, but take your time, 2 minutes. All right, let's read the description. A process that lead to genetic variation. A process that lead to genetic variation. Remember, we are in the chapter or the topic of meiosis. So the context is meiosis, okay? Random arrangement of chromosomes. Does that lead to genetic variation? If you've watched the lessons, yes. So you don't eliminate that one. So option B is crossing over. Does that lead to genetic variation as well? Yes. So we have not eliminated both of them. So meaning option A is viable and option B is viable. So what do we write? We don't just write both. We write both A and B for 2 marks. Following question. The importance of meiosis. Remember, meiosis has 3 main importances or significances. Okay, let's see if the options fall under the 3. Production of gametes. Does meiosis produce gametes? It does. So we do not eliminate that. Halving of the chromosome number. Does meiosis halve the chromosome number? Yes, it does. Remember, we said that it reduces the number of chromosomes from diploid to haploid. So that is also correct. So both of them are correct. We don't just write both. What do we write? We write both A and B. Following question. Cytokinesis takes place. Cytokinesis takes place. So before we look at the options, cytokinesis. What is cytokinesis again? We say that cytokinesis is the division of the cytoplasm. All right? When we have one cell, then we divide the cytoplasm. So when we divide the cytoplasm, it means that we have two daughter cells. Okay? So let's look at the option. Cytokinesis takes place, telophase 1. Could that be correct? Yes. Remember, in telophase 1, we are dividing one cell into two daughter cells. So we divide the cytoplasm from being 1 to 2. So we don't eliminate telophase 1 because we are confident that it is correct. And then option B, telophase 2. Does cytokinesis take place during telophase 2? Absolutely. Remember, the two daughter cells that we start with during meiosis 1 are going to divide. Okay, they are also going to divide to produce two daughter cells each, and then we have a total of 4. So both option A and option B are correct. So what do we write? The correct answer is both A and B, and then for 2 marks. The next question. Study the diagrams below which illustrate some phases of meiosis 1. Study the diagrams below which illustrate some phases of meiosis 1. Now, we can't just pass through this instruction. There is something very important. There's a huge clue here. So, these diagrams represent only phases that are included in meiosis 1. So this brings context to the question, brings context to how you're supposed to be thinking in order to derive your answers. So none of your answers are going to include meiosis 2. They should only be derived from meiosis 1. Given the context of the question. Okay, so let's study the diagram first before we even look at the questions. Remember, that's what you must do when you are answering these questions. All right? Okay, so diagram 1, diagram 2, diagram 3, and then we have unlabeled parts, part A as well as part B. So we are supposed to identify the phases in each diagram, and we are supposed to label the parts A and part B to get some context before we even look at the questions. Let us check. So diagram 1. Remember, your job is to be able to just look at the cell, the activities in the cell, and tell which phase does each diagram belong to, depending on what is happening within the cell. Okay, guys? So that's why it is very much important for you to know how the chromosomes are acting or behaving, and what are the activities that are happening within the cell to be able to identify that particular phase. Okay, let's start with diagram 1. What is happening in diagram 1? What we see is that whole chromosomes, not chromatids, whole chromosomes are being pulled to the opposite poles of the cell. So we assume that the spindle fibers, which are there, are contracting. Remember, the whole chromosomes are pulled to opposite poles of the cell because of the contraction, the pulling effect of by the spindle fibers. And then let's identify part A. Something that I must tell you that I forgot to tell you in the past: examiners will never include unlabeled parts such as part A and part B. Right? Would never include them if they are not going to ask something about them. They're either going to ask you directly to label them or to identify them, or they can ask you an indirect question relating to that particular part that is included, that is not labeled. So before you even look at the questions, guys, don't let your mind work in reverse. Before you even look at the question, label. It's not only for meiosis, but for any structure that you are given. Please label them first. Know what the parts are for you to be able to answer the questions. Don't go to the questions. Study the diagram first. Okay. So we said diagram 1 is anaphase 1. Why? Because whole chromosomes, as we can see, are being pulled to opposite poles of the cell. Okay? Then diagram 2, let's check what's happening. Diagram 2. In diagram 2, we see that chromosomes are aligned at the equator, but in homologous pairs. So when does this happen? This happens at metaphase 1. And then structure B, as you can see over there, is the spindle fiber or the spindle thread or the microtubule. And then diagram 1, we forgot. Depending on the context of the question, what I see here is that the label line, please make sure that you check directly what the label line is indicating to. So I am seeing a chromatid. Okay? I am seeing a chromatid at face value. However, depending on the context of the question, the answer might be different. But what I see for now is that this is a chromatid. Right. Now, diagram 3. Let's check what's happening in diagram 3. All we see is that the chromosomes are aligned in homologous pairs, but there is something that is happening there. We see that the chromatids of the homologous chromosomes have overlapped. Okay? Or they have overlapped, as you can see over there. And which process within meiosis do we know happens when the chromatids of the homologous chromosomes overlap? And that is called crossing over. And when during meiosis does crossing over take place, it takes place during prophase 1, not prophase 2, prophase 1. All right. Another thing that we see is that the nuclear membrane has not disappeared yet. Okay? Remember, during prophase 1, the nuclear membrane begins to disappear. It does not disappear altogether, but it begins to disappear. Okay? That also happens during prophase 2. But what can tell us here that, or indicate to us, to confidently say this is prophase 1? Two things. Chromosomes are arranged as in homologous pairs, number 1, and number 2, the crossing over is taking place. All right? So that's why we can confidently say that this is prophase 1. We have studied the diagram. Now we are confident and ready to answer the questions. Obviously, the first question is going to be label parts A and part B. So you would have labeled this in your question paper, both parts. You would have taken notes in your question paper. All you have to do now is to transcribe to your answer book. So part A is a chromatid, like we said, and then part B is the spindle fiber, or you can say spindle thread, or microtubules will also be correct. Question 2.2. The diagrams above are not. They are not placed in the correct sequence. Use the diagram numbers to write down the correct sequence in which the part of the process of meiosis 1 takes place. Okay? They say there, use the diagram numbers, not the names of the phases. Use the diagram numbers, guys. So please answer according to the given context. They say use the diagram numbers. Don't name them. Use the diagram numbers to write down the correct sequence in which part of the process of meiosis 1 takes place. So we know that meiosis 1 has 4 phases. However, here they've included only 3. Okay? So this is why it was very important for us to study the diagram prior to even looking at the questions and to make notes. We said that remember, the sequence of meiosis goes like this. We said we abbreviate it as P mat. P mat. So prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay? Now, we say that this is diagram 1. We say that it is anaphase 1. Excuse me. We say that diagram 2 is metaphase 1. And then diagram 3, we had said that it is prophase 1. Okay? So in this sequence over here, prophase 1, metaphase 1, anaphase 1, telophase 1, we check the sequence. Okay? So what comes first? What comes first is diagram 3, because it is the first phase of meiosis, prophase 1. And then according to this sequence, metaphase 1 is next. So we are going to have diagram 2. All right? And according to this sequence, anaphase 1 is next. and then we are going to have diagram 1. Let us check if that is the correct answer. Correct. So, prophase 1 first, metaphase 1 next, and anaphase 1 last. So we don't have telophase 1 depicted there. The last question. Give 2, okay? Give 2. Give 2 observable reasons why the phases in the diagram are not part or are part of meiosis 1. Give 2 observable. So when they say observable, they mean that the ones that we can observe in this diagram, the ones that we can see. Give 2 observable reasons why the phases in the diagram are part of meiosis 1. So they are telling you that they are part of meiosis 1. What you are required to do is to give 2 reasons that are observable, reasons that are seen in this diagram, to justify or to account for the fact that they are indeed part of meiosis 1. Let us look at the answers. What do we see from diagram 1? In diagram 1, whole chromosomes are pulled to opposite poles of the cell. Whole chromosomes. Just a word of advice again. Please learn to answer the questions directly and not indirectly. Some learners, or candidates, should I say, some candidates at the marking center, when we mark the final papers, we see them answering indirectly. They would say, for example, chromatids are not pulled to opposite poles. Chromatids are not pulled to opposite poles. Now, you've answered the question, but not directly. You should be direct. If chromatids are not pulled to opposite poles of the cell, what is being pulled to opposite poles of the cell? Okay? Answer the questions directly, please. So whole chromosomes are pulled to opposite poles of the cell. That is what we see in diagram 1. Hence, we justify to say this is anaphase 1, not anaphase 2. If it was anaphase 2, then chromatids will be pulled to opposite poles of the cell. What can we observe in diagram 2? In diagram 2, chromosomes are arranged at the equator of the cell in homologous pairs. In homologous pairs, or in 2 rows. In 2 rows, row 1 and row 2. That will also suffice. Okay, what can we observe in diagram 3? Like we said earlier, chromosomes are arranged in homologous pairs, or there's one additional thing that we can see there. Crossing over is taking place. Right? So that's why we can confidently say diagram 1, diagram 2, diagram 3, what can we identify in each diagram to confidently say that this is the process of meiosis 1. Okay? But we only answer 2, the first 2 that are correct. The next set of questions. The diagrams below show chromosome pair 21 in the nucleus of a cell of the ovary of a woman. The chromosomes are involved in a process that takes place in a phase of meiosis. Okay, so let's analyze the statement. The diagrams below show chromosome pair 21. So this is the pair of chromosomes, pair 21 in particular, in the nucleus of a cell of the ovary of a woman. Okay? The chromosomes are involved in a process, so they are telling us that it is involved in a process without revealing which process it is that takes place in a phase of meiosis. In a phase of meiosis. So there's a few things that we need to look at. Chromosome pair 21, that is our clue. The nucleus of a woman, in a cell of the ovary of a woman. Cell of the ovary, not ovum, ovary. Right? Remember what we said about somatic cells and gametes? Any other cell of the body besides an ovum or a sperm is a somatic cell, meaning it has two sets of chromosomes with 46 chromosomes in total. It's a cell of the ovary, or that makes up the ovary of a woman. The chromosomes are involved in a process, so it's our job to identify the process that takes place in a phase of meiosis. All right? Let's try and check which phase and what process the statement is referring to. Okay, so we have diagram X, Y, and Z. In diagram X, we see a homologous pair of chromosomes, and from the statement they said it's chromosome pair 21, a homologous pair of chromosomes. And then what is it that we can see in this diagram? We see that there are different colors, or there are different shades of the chromosome. This chromosome over here is a majority black, and only that part is white. This one is a majority white, and only that part is shaded black. Okay, so remember the shades of colors, or the colors, represent different combination of what? Different combination of genes. All right. This black is from this part of this chromosome, and this white is from that part of the chromosome. So it means there was an exchange of genes at that point over there. There was an exchange of genes at that part over there. Okay? So it has already taken place. And which process is involved with the exchange of genes? We know that as crossing over. So the process that the statement might be referring to might be crossing over, depending on the context of the question. And then diagram Y, we see that it is the same homologous chromosomes. Okay? It is the same homologous chromosomes. So label A will be homologous chromosomes. But they have not, as you can see, exchanged genes yet, meaning crossing over has not taken place yet. And then diagram 2, we have structure B. Okay, structure B is in the center of this one chromosome. So structure B is definitely the centromere, because it is in the center and it is holding the two chromatids of this chromosome together. What else do we notice there? We notice that the chromatids of the homologous chromosomes have overlapped there. So this would be our chiasma. All right. So the process that they are referring to, I have reason to believe that it is crossing over. And they say that these three diagrams are showing a phase of meiosis. So all three of these diagrams are involved in one phase. And where, or rather when, does the process of crossing over takes place? It takes place during prophase 1. Okay, let's now look at the questions. Of course, the first question is going to be give labels A and B. So the answer, like we have said, A is homologous chromosomes, or you can say a homologous pair of chromosomes, and then B, it is the centromere. So we were right. Question 232, excuse me. Just like the question on the previous set of questions, rearrange the letters X, Y, and Z to show the correct sequence in which the events take place in this phase, X, Y, and Z. So let us go back to the diagram. So what do we see? Diagram Y is before crossing over takes place, as you can see. So this would be our first. And then diagram Z is during crossing over. So that would be the second diagram in the sequence, and diagram X would be the last step after crossing over has taken place. So let's see if we are correct. So our sequence is going to be Y, Z, X. Let's see, Y, Z, X for 1 mark. That is correct. Following question. Explain why the chromosomes in diagram X and diagram Y are different in appearance. Firstly, action verb, explain. Hence, marker location, 3 marks. Explain why the chromosomes in diagram X and diagram Y are different in appearance. So in diagram X, as you can see over there, let me just erase. As you can see over there, are not the same as in diagram Y, as you can see over there. So in this diagram, one chromosome is all black, another chromosome is all white. This chromosome is a majority black with a touch of white, and in that chromosome, a majority white with a touch of black. Why is that the case? Of course, it's because of crossing over. But how do we articulate that for 3 marks? Right? In diagram Z, chromatids of the homologous chromosomes overlap, 1 mark. There's an exchange of genes, 1 mark at the chiasma. And then for the last mark, which results in each homologous chromosome having a different combination of genes, such as in diagram Y. All right? Is that the last question? Yes, that was the last question here. Moving on to the next. Okay, guys, this is the last of our questions. It is question 3. And the question reads, the diagrams below represent 2 phases of meiosis in an organism. So these diagrams represent, let's just underline what's important, 2 phases. We are not told which phase it is, and then in an organism, therefore they don't say it is a human being. Okay, so remember what we usually do, we study the diagram and then we look at the questions. Okay, that's what you should do in the exam as well. All right, let us firstly start by identifying the diagrams. So your point of departure is you check what is happening in the cell before you can determine which phase of meiosis that diagram represents. Okay, so what we see here is that the spindle fibers are attached to the centromeres of unreplicated chromosomes. And what we can see is that we have unreplicated chromosomes or chromatids that are moving to the opposite poles of the cell. Okay, and we know that when the chromosome splits and chromatids and or unreplicated chromosomes move to the opposite poles of the cell, diagram 1 would therefore represent which phase? It would represent anaphase 2. We have that structure over there that is unlabeled. So that structure there is at the pole. Therefore, it is the centriole. Remember, the centriole is at the pole. Remember that rhyming sound. In diagram 2, let's check what is happening. We have single chromosomes that are aligned, or individual chromosomes that are aligned at the equator of the cell. Remember, the word equator just means the middle of the cell. And spindle fibers are attached to the centromeres of the individual chromosomes. Now, which phase is that? We know that we have two phases where chromosomes are aligning at the equator. If they were aligning at the equator in homologous pairs, it would be metaphase 1. But here they are aligned at the equator singly or individually, and that is metaphase 2. Okay, so we have that structure in the middle or at the center of the replicated chromosome. What is that structure called? That is called the centromere. And then we have that structure that is produced by the centriole, and it is attached to the centromere. That structure is known as the spindle fiber or spindle thread. And we have structure D. Structure D is a replicated chromosome. Okay, now let's look at the questions. Of course, the first question is going to be identify and state one function of part A and part B respectively. Now look at this question, guys. It says identify and state one function. An error that we mostly see at the marking center when we mark the NSC Life Sciences exams at the end of the year, a lot of candidates usually miss this and part. Okay, when it says and, remember it's an instruction. You get a mark for identifying and you get a mark for stating one function of these two parts. Okay, and look at the mark allocation, it's 4. So if the mark allocation is 4 marks and then you have 2 parts, on those 2 parts you have to identify and state one function. It means that it is going to be 1 mark for identifying and 1 mark for the function. So please do not selectively read the questions. Read the question in its entirety first and underline the things that are important when you read it for the second time, guys, or else you are going to lose marks. Okay, now the word respectively means separately or individually. Okay, therefore you have to identify and give a function of part A, and then you have to identify and give a function for part B to get a total of 4 marks. Let's check part A. What is part A? Remember, identify. Part A is at the pole. Therefore, part A is the centriole. Okay? And we know that the function of the centriole is to produce spindle fibers or spindle threads. Let's check if that is correct. Part A is precisely that, the centriole, and it produces spindle fibers. Let's look at part B. There is part B, okay? So part B, look at the label line. It's not on the entire chromosome, okay? Please follow the label line carefully. It is on that circle structure in the center of the chromosome. Excuse me, pardon me for that. It is showing that structure in the center of the chromosome. We know that as the centromere, okay? And the function of the centromere is that it holds two chromatids of replicated chromosomes together. Remember, this chromosome has two chromatids, therefore it is replicated. Both of the chromatids are held together by a centromere. Therefore, part B is the centromere, 1 mark, and it joins two chromatids of a replicated chromosome together. The word replicated in this case is optional. Even if you had written it joins two chromatids of a chromosome, it would suffice, okay? But the most correct answer is replicated chromosome, but because it is written in brackets, it means that it is optional. Whether that word is there or not, you will be credited a mark, okay? But my advice is to write it joins two chromatids of a replicated chromosome together, because another examiner might want that word to be included. All right, question 3.2. Explain what happens to part D in the next phase of meiosis. Part D we have identified as a chromosome, a replicated chromosome, a chromosome that has two chromatids. So our job is to explain what happens to the replicated chromosome in the next phase of meiosis, not this phase of meiosis, in the next phase of meiosis. Now, I have included 1 mark there. There's something wrong with the mark location. If it is an explain question, it cannot be 1 mark. It's supposed to be 2 marks. So excuse me for that. All right. Now, here, remember our abbreviation. PMAT. PMAT. Remember that abbreviation is going to help us a lot. So this abbreviation does not only help you to identify the different phases or memorize the different phases, it also helps you to check the order in which they appear. I'm sure we can agree that this cell is showing metaphase 1 because individual chromosomes are lined at the equator, okay? So this is where we are so far. So diagram 2 is showing metaphase 2, okay? It is showing metaphase 2. Therefore, the next phase of meiosis would be anaphase 2. So what the question is requiring us to do is to explain what particularly happens to the replicated chromosome structure D during anaphase 2. Okay? So the answer to that is... Okay, before we look at the answer, what happens during anaphase 2? The spindle fibers will contract. Okay? So remember, we said that the word contract means pull. So this spindle fiber is going to pull towards that direction. This spindle fiber pulls towards that direction. This spindle fiber pulls towards that direction. Remember, it is pulling towards the direction of the opposite poles. Okay? And that spindle fiber will pull to that direction. Now, if you have a spindle fiber that is attached on this side of the chromosome and another spindle fiber that is attached to that side of the same chromosome, and they are pulling at opposite directions, it means that this chromosome, replicated chromosome, is going to split in half. Okay, and then we are going to have a situation where this chromatid or unreplicated chromosome will move to that pole of the cell, and that chromatid or unreplicated chromosome will move to that pole of the cell. Okay, let's see if that is the answer. It's going to split 1 mark or 1 tick, and the chromatids are pulled to the opposite poles of the cell. 1 tick. Guys, remember, I have made a mistake and included 1 mark for an explain question. It's not supposed to be 1 mark. It's supposed to be 2 marks there, and it's supposed to be 2 marks there as well. Okay, excuse me for that error. The following question: give the number of chromosomes present in sub-question A of question 3.3, the original parent cell in this diagram. The original parent cell in this diagram. Remember, we don't want the pairs of chromosomes. We need the number of chromosomes. Remember what we said. We said that meiosis works like this. We have an original cell. The original cell is always diploid. Okay? It is going to undergo meiosis 1 to produce 2 haploid daughter cells. Okay? And then that is meiosis 1. Excuse me, that is meiosis 1. Okay? I'm going to write M1. That is meiosis 1. And during meiosis 2, these daughter cells are now haploid. They contain half the number of chromosomes. All right? Now, these daughter cells will divide each to produce 2 daughter cells. Each are to produce 2 daughter cells. At the end, we are going to have 4 daughter cells. Okay? Now, that would be meiosis. That would be meiosis 2. And these 4 daughter cells are haploid each. Okay? Now, let us look at these 2 phases. These 2 phases of that are represented in these 2 diagrams are undergoing meiosis 2. Okay? Are undergoing meiosis 2. This is metaphase 2, and this is anaphase 2. Okay? Now, if they are undergoing meiosis 2, it means that they are haploid already. And as you can see on diagram 1, we have how many chromosomes? We have 2 chromosomes at a phase at meiosis 2. Remember what we said? We said that cells in meiosis 2 are already haploid. Okay, now the question is asking us to provide a number of chromosomes. A number of chromosomes at the original, in the original cell. So, to move from a haploid cell to a diploid cell, we multiply the haploid number by 2. So 2 times 2 is going to give us how many chromosomes in this original cell? 4 original chromosomes. Okay? Remember that the cell under meiosis 2 is haploid. The cells are always haploid. And then the cell under meiosis 1 are always diploid. So here we have a cell at meiosis 2. The question is asking us to give the chromosome number at meiosis 1. So we multiply that by 2. That means that the original cell had 4 chromosomes. Okay, let's check if that's correct. That is very correct. Moving on to the following question, or rather the following sub-question. So sub-question B of question 3.3. A human cell, Give the number of the chromosomes present in a human cell in the same phase of meiosis in diagram 2. So diagram 2 is showing metaphase 2. Okay, now the number of chromosomes in a human cell, the diploid number in humans is 46 chromosomes. The diploid number is 46 chromosomes. It means that the haploid number in a cell undergoing meiosis 2, remember the cells under meiosis 2 are always haploid, meaning we divide 46 by 2, which is going to give us 23. Remember, we said to move from a diploid number to a haploid number, we just divide by 2. And to move from a haploid number to a diploid number, we multiply by 2. Okay, hence we have 23 chromosomes. The following question is a drawing question. Guys, you will always be asked somewhere to draw something, okay? So here you are supposed to draw a labeled diagram, guys. A labeled diagram. A labeled diagram of what? Of a single cell that results in the next phase. That results in the next phase of diagram 1. Let's go back to our abbreviation. PMAT. PMAT. So this would be PMAT 2. Prophase 2, metaphase 2, anaphase 2, as well as telophase 2. Okay, so when we look at this cell, we have already identified that in diagram 1, chromatids or unreplicated chromosomes are pulled to the opposite poles of the cell, and that happens during anaphase 2. Okay, so the next phase from anaphase 2 will be telophase 2. So we are supposed to draw a label diagram of a single cell, just one cell, that results, meaning after the next phase, which is telophase 2 of diagram 1. So we are looking to draw a cell after telophase 1, okay? And then the question says a single cell. We know that it will be 2 cells that result from one daughter cell, okay? But the question is restricting us to only a single cell, okay? Now in life sciences, when you are asked to draw anything, guys, you must provide a heading. That is your point of departure. And please write down the heading before you draw, because often many learners forget to write the heading because they leave it as the last thing that they do. Now, to not forget a heading, write it down as the first thing that you do. Now, what is our heading supposed to do? What is our heading supposed to be? Your heading will not be a labeled diagram of a single cell that results in the next phase of diagram 1. That is too vague, guys. That is too vague. Right? We are supposed to draw a single cell after telophase 2. Okay, after telophase 2. So our heading, a cell during telophase 2. Actually, this is wrong. This is a wrong title. It's not supposed to be during telophase 2. It's supposed to be after telophase 2. Okay. Now, you can't draw a cell without including a cell membrane. So you may draw a circle that represents a cell membrane, and then we have a void in that cell membrane, and we know that void or that cavity would be filled by the cytoplasm. And then you draw an additional circle that represents the nuclear membrane. Okay, remember, after telophase 2, the nuclear membrane reappears around the chromosomes. Okay, that's why we are including a nucleus. Every other phase should not include a nucleus. Only during prophase 1, when it begins to disappear, and after telophase 2. Okay, so there is our nuclear membrane. And then our chromosomes, either you include, remember, there's supposed to be two cells that result from the cell. Either you include these two chromosomes in your diagram shaded like that, or you include those chromosomes in your diagram shaded like that. Okay, they must be shaded like that. And then what else can we include as labels? So that would be unreplicated chromosomes, not replicated chromosome. This should be an unreplicated chromosome. But because it is included in brackets, it means that it is optional. You writing chromosome is perfectly fine. Okay? And then the following label, you can include the centromere. It's fine. And then outside of the nucleus, you should also include the centrosome. Okay? So as a guide to mark this answer, I will be providing the marking guideline. H is for the heading, the correct heading, that is a heading that does not say a drawing of a single cell in the next phase of diagram 1. No. You must include what the next phase is. And the next phase is telophase 2. Okay? Telophase 2. All right? And it's not during, it is after telophase 2. You are showing a single cell after telophase 2 to qualify for that 1 mark. And then the next thing, the correct type of drawing. You drew only one cell because the question says a single cell. Okay? Your cell must include 2 unreplicated chromosomes, either these 2 shaded like that or those 2 shaded like that, to qualify for the mark for the correct type of drawing. So it should show 2 unreplicated chromosomes, and in the shades, either those or those. Okay, to qualify for that 1 mark. And then any 3 correct labels. So any 3 correct labels. So if you have many labels and some of them are wrong, the marker will ignore the wrong ones, and they are obliged to mark the ones that are correct. Okay, so any 3 labels on the diagram, the marker, you'll see a tick, maybe the marker will choose that one also be a tick. Only 3. The maximum is 3 according to the marking guideline. I would like to caution you now, guys. The first thing that I've already cautioned is the fact that, please, when you're asked to draw anything, please include a heading for that particular structure that you are drawing. Secondly, the second thing that I would like to caution, the question says draw a labeled diagram. Please do not selectively read, guys. A labeled diagram, meaning a diagram with labels. Even if the instruction did not say labeled diagram, it just said draw a diagram, you must check the mark allocation. The mark allocation is 5 marks. You must ask yourself in your mind, you must ask yourself, where do these 5 marks come from? You know that always 1 will be for the heading. you'll see the marker writing H and then a tick or a cross. The other mark will always be, for the correct type of drawing, another mark. Okay, so we've already used up 2 marks. The other 3 marks should be for the labels. So when you're asked to draw a structure or a cell, please include headings, include labels. Even if the diagram does not say labeled diagram, even if it says diagram, please include headings. Labels, excuse me. Please include labels, especially when the mark allocation is as expensive as 5 marks. You cannot afford to lose these 3 marks because of ignorance. I'm not saying you are ignorant, but the act of not including labels is ignorant. Okay, guys? Now, for the last question, do we have another question? Yes. Provide one visible reason why the cell represented by diagram 1 is not a human cell. So there, it's not supposed to say when. There's so many errors in the slide. Sorry, guys. Maybe I don't know what was wrong with me. But, sorry, it's supposed to say why, not when. Okay, why is the cell in diagram 1 not a human cell? So you're supposed to check the cell at diagram 1 and tell why it is not a human cell. Okay, what do we know about a human cell What do we know about a human cell that is undergoing anaphase 2? What we know is that there's supposed to be 23 chromosomes or chromatids that are moving to that pole, and 23 chromatids or unreplicated chromosomes that are moving to that pole. Okay? But when you check this diagram, there are only 2 chromatids moving to opposite poles of the cell. Okay? I do not have space. I did not create space. We have reached our capacity for writing. But please make a note of this. Excuse me. The answer to this question will be 23 chromosomes, unreplicated chromosomes, or 23 chromatids should move to the opposite poles of the cell. 1 tick. However, in diagram 1, only 2 chromatids or unreplicated chromosomes are moving to the opposite poles of the cell. With that, we have reached the conclusion of our penultimate lesson, lesson 3 on the lesson series of meiosis. Okay, guys, I hope you enjoyed this lesson. Please do me a small favor. It might be a small task for you or a small favor for you, but it will mean the world to me and the growth of my channel. Please do react to this please do react to this video. Comment any contribution that you might have, any question that you might have. Hit the subscribe button, and please do share the link to this video, as it will help my channel grow. Okay. The next lesson will be a lesson on abnormal meiosis. That will be the final lesson in the lesson series of meiosis. As usual, thank you so very much for watching. I appreciate you, and I'll see you next time. Goodbye.